Hi everyone, welcome to our session. Now we are discussing about immunofluorescence microscopy and fluorescence recovery after photobleaching. Myself Anjali PT, 2nd MSc Biology, St. Mary's College, Thrissur. Now we are discussing about introduction of immunofluorescence. Immunofluorescence is a powerful technique that utilizes fluorescent labeled antibody to detect specific target antigens. We are using a dye, fluorescein. Fluorescein is a dye which emits greenish fluorescence under ultraviolet light. It can be tagged to immunoglobulin molecules. Immunofluorescent labeled tissue sections are studied using a fluorescence microscope. Then immunofluorescence is a technique allowing the visualization of specific protein or antigen in tissue sections by binding a specific antibody chemically conjugated with fluorescent dye such as fluorescein isothiocyanate FITC. FITC is the short form of fluorescein isothiocyanate. The specific antibodies are labeled with a compound that makes them glow and apple green color when observed microscopically under ultraviolet light. Then we are telling about fluorescence. Fluorescence is the property of certain molecules or fluorophore to absorb light at longer wavelength when it's illuminated by light of a different wavelength. Then we are telling about fluorescence emission. What is fluorescence emission? The incident light excites molecule to a higher level vibrational energy. As the molecules return to the ground state, the excited fluorophore emit a photon that is called fluorescence emission. This is the examples of fluorescence dye. One is fluorescein and second one is rhodomine. Then principle. Immunofluorescence utilizes the specificity of antibodies with fluorescent dye to recognize their antigen and therefore allows visualization of the distribution of the target molecule through a fluorescent dye with a fluorescence microscope. Then we are telling about immunofluorescence staining. Immunofluorescence staining, two types: direct immunofluorescence and indirect immunofluorescence. Direct immunofluorescence is the staining in which the primary antibody labeled with fluorescence dye, and indirect immunofluorescence is the staining in which a secondary antibody labeled with fluorochrome is used to recognize a primary antibody. This is the difference between direct and indirect. Direct staining in which the primary antibody labeled with the fluorescence dye, and second one is indirect immunofluorescence. Staining in which a secondary antibody labeled with fluorochrome is used to recognize a primary antibody. Yellow color is antigen and bluish color is primary antibody and ash color is secondary antibody and green color fluorophore. Then we are discussing about direct immunofluorescence. This is the picture of direct immunofluorescence. It is the simple and very common procedure. Antigen is fixed on the slide. Fluorescein labeled antibodies are layered over it. Slide is washed to remove unattached antibodies examined under ultraviolet light in an fluorescent microscope. The site where the antibody attached to its specific antigen will show apple green fluorescence. The color is apple green fluorescence. That is direct immunofluorescence. Then advantages of direct immunofluorescence. This technique has several advantages over indirect immunofluorescence because the direct conjugation of the antibody to the fluorophore. This reduces the number of steps in the staining procedure, making the process faster and can reduce background signal by avoiding some issues with cross-reactivity. Then direct immunofluorescence uses. It is used to detect viral parasitic tumor antigens from patient specimens or monolayer of cells. Another one is indirect immunofluorescence. Indirect test is a double layer technique. The unlabeled antibody is applied directly to the tissue substrate, treated with a fluorochrome conjugated anti-immunoglobulin serum. That is indirect immunofluorescence. This is the picture of indirect immunofluorescence. We can see unlabeled antibody. The blue color is fluorochrome labeled antigen. Then advantages of indirect immunofluorescence. It gives an amplification effect and it requires only one labeled antibody to identify many proteins. Indirect immunofluorescence is used to detect autoantibodies such as anti-nuclear antibodies in autoimmune diseases and to identify the class of a given antibody by using fluorescent antibodies specific for different immunoglobulin isotypes. This is some of immunofluorescence images. This is immunofluorescence image of Cryptosporidium parvum oocytes, and another one is Haemophilus influenzae. Then next topic is fluorescence recovery after photo bleaching, FRAP short form. What is photo bleaching? Photo bleaching is a photochemical destruction of a fluorophore. In microscopy, photo bleaching may complicate the observation of fluorescent molecules since they will eventually be destroyed by the light exposure necessary to stimulate them into fluorescence. Photo bleaching may also be exploited to study the motion and or diffusion of molecules, for example via the FRAP or FLIP technique. FRAP, fluorescent recovery after photo bleaching. Then we are telling about FRAP. FRAP, fluorescent recovery after photo bleaching. In FRAP experiments, fluorescent molecules are irreversibly photo bleached in a small area of the cell by high-powered focused laser beam. Subsequent diffusion of surrounding non-bleached fluorescent molecule into the bleached area leads to a recovery of flow which is recorded at a low laser power. Then the basic apparatus comprises an optical microscope, a light source, and some fluorescent probe. Fluorescent emission is contingent upon absorption of a specific optical wavelength or color which restrict the choice of lamps. Then source of light. Most commonly a broad spectrum mercury xenon source is used in conjunction with color filter. The light source is focused onto a small patch of the viewable area. Then the fluorophores in this region receive high intensity illumination which causes their fluorescence lifetime quickly elapse. Now the image in the microscope is that of a uniformly fluorescent field with a noticeable dark spot. The fluorescent probe will diffuse throughout the sample and replace the non-fluorescent probes in the bleached region. The diffusion proceeds in an ordered fashion analytically determinable from the diffusion equation. D is the diffusion constant, and diffusion constant D equal to W square divided by 4T 1 by 2. W is the radius of the beam and T half. T half is the time required from the bleach spot to recover half of its initial integrated intensity. Once more, I will tell you the diffusion constant D can be simply calculated from D equal to W square divided by 4T half, where W is the radius of the beam and T half is the time required for the bleach spot to recover half of its initial integrated intensity. This is the picture, the red top view and side view of the fluorescence. Then principle of Frap, fluorescence recovered after photo bleaching. First, the bilayer is uniformly labeled with a fluorescent tag. Bilayer is uniformly labeled with a fluorescent tag. This label is selectively photo bleached by a small 30 micrometer fast light pulse. The intensity within this bleached area is monitored as the bleached dye diffuses out and new dye diffuses. Eventually, uniform intensity is restored. This is the principle of Frap. Then, applications of Frap in living cells. Frap can be used to address a number of questions about protein localization, dynamics, and interactions with other components in living cells. The mobility of molecules within specific cell compartments has been visualized as membrane continuity. Then Frap is a technique to study molecular dynamics, is majority used to study the membrane fluidity and study the mobility of membrane protein, and also the studying of cytoskeletal dynamics and intercellular transport. Thank you watching for this video.